Okay, guys, we're nearing the end. Hopefully, this might be the end of it, but we'll see. Um, Yada, yada, yada. There's a lot to read in here. Let's get right to the pictures, okay? Let's get right to the pictures. And I really like this picture a lot. This is an example of an organism that would have a diploid number of 2, okay? It only has 2 different chromosomes, 2n, okay? 2n equals 2. So it has the one and the other, okay? Um, yes. Okay. Now, what happens is since you have your 2n number, oh, you know what? Yes. Since your 2n number is 2, okay? Then your n number is going to be 1. What happens? Here's the DNA, okay? Yes, it's all scrunched up, but think of it as all stretched out. It has to replicate and then shrink down. Here's that chromosome that it got from its dad. Here's the chromosome it got from his mom. In the first phase of meiosis, the chromosomes, the homologous chromosomes, and remember we can go back to a karyotype, will all line up in their pairs, like pair number 1, pair number 2, if this was a human. We'd have pair number 1, 2, 3, 4, 23, right? They will divide, but notice they're still replicated, okay? When they divide, they've already reduced the chromosome number. In fact, they call meiosis 1 the reduction division, because as that cell divides, we reduce the chromosome number. In the second one, they call it the mitotic division because it looks just like mitosis. If you had started out with this cell and it went through mitosis, every single chromosome would line up right in the middle, and then boom, they'd split as single chromosome. The replicated chromosome would split into 2 single chromosomes, okay? So let's look at this picture, okay? And this is on the side. I could turn it this way, but I'm not going to, okay? Here is a cell, and if we count how many chromosomes are in it, we got 1, 2, 3, 4, 5, 6. So this has a diploid number, okay? 2n equals 6, okay? Now, we'll get into what's going on with this tangling up here in a moment. But notice how the chromosomes are lined up. Sometimes the dad one's on this side, sometimes the mom one's on this side. See, dad one's on that side, mom one's on this side. Okay? See what's happening here? Dad's on this side, mom's on this side. But we have, like if this is chromosome number 1, pair number 1, pair number 2, pair number 3, they're lining up as homologous chromosomes. And they're splitting still replicated. Okay? Can't keep them replicated. That's our first division. That's the reduction division. So from here to here, this is all the reduction division. We've reduced the chromosome number now in half. And how can I prove it? We had 6 here, but how many do we have here? 1, 2, 3. 1, 2, 3. 1, 2, 3, 4, 5, 6. We've reduced the chromosome number when we divide it. But the problem is we can't leave them replicated. If they were single, we'd be done, but they're not. So in the mitotic division, and they call it the mitotic division because it's just like mitosis, okay? Now they line up right on the equator, single file, not in their pairs. Notice the equator was turned to the side because cells don't stay put. Now they're lined up right on. And now notice that we're getting single chromosomes. And notice 1, 2, 3 single chromosomes. 1, 2, 3 single chromosomes. 1, 2, 3, 1, 2, 3. Okay, we've now gone from 2n to here. And I also like to do it like this, 2n that are replicated still. Okay, here we have n, which equals 3, but they're still replicated. And now we have n that are single, and there's 3 apiece. Okay, so it's very important to see the difference between the replicated chromosomes and the single chromosomes. We need to have single chromosomes, and we need to have half the chromosome number. 1 chromosome from each of those homologous pairs. So they had 3 homologous pairs. Do they have 1 from each one? Yes, so it all checks out. But what was this little tangle that was going on? And this is something I definitely know that KB and Balkis and myself and Metzger all talk about. Because during that first division, sorry that I moved that camera there, during that first division, that reduction division, what happens sometimes is when they're pairing up with their homologous pairs, they kind of do a little switcheroo, okay? They kind of like exchange some parts. They're all like tangled apart, and they say, "Hey, I'll have your like A here, and you have my B. Oh, okay, I'll have your B and C, but you can have my B and C." Okay, so what do I mean by that? Those are certain genes that they're saying tit for tat, kind of. Okay, whatever gene is here, it's switched up here. And so what we see is we see more variety going on here. So we have this chiasmata, okay, or this crossing over. I like to call it crossing over. It's just an easier word to pronounce too, or two words. Crossing over happens. We have a little switcheroo that mixes up the genetics. See, like when we look at these chromosomes, this mom chromosome has a little piece of dad chromosome on it. Same here. These dad chromosomes have little pieces of mom chromosome on them. And it happened way back here when that little switcheroo kind of happened, when they were lining up as homologous pairs. That just adds to the diversity, because just the way they lined up was one way, because like we were going to get some mom things and some dad things on this side, some mom things and some dad things on this side. But now we even have more diversity because they have little pieces of mom things with dad things. Okay. And this is why we get like so much diversity in just like one family. Okay, so we have not only the alignment of the homologous pairs, and that's what we call independent assortment. That's what's going on here. So this here, okay, that's independent assortment. Independent assortment. Not only do we have independent assortment, or the aligning of them, bringing out diversity, but we also have crossing over, bringing about diversity. So when, say, this is like sperm that were all made here, they're going to grow little tails eventually, right? These sperm are all carrying quite a variety. Not every sperm is alike. Okay, so that's what's kind of interesting here. And it leads us into, oh, blah, 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 blah. You can read this at another time. It talks about all the phases that we just did, but focus on the things we said. Oh, why do I type all that stuff up? Let's move on. Watch this video. Definitely. Good video. Very, very good video. If you didn't watch any of the other ones, definitely watch this one, because it will help clear up meiosis. But here, we're going to compare meiosis to mitosis. Remember, mitosis, we only get one division. Okay, they all line up single file and divide, and they single, and they're going to be all the same. Okay, here though, they line up in pairs. They split once, then they have to split again. Yes, here we end up with two cells that are both diploid. Okay, begin with diploid cells. Remember, it begins as 2N that are replicated, but it ends with 2N that are single, 2N that are single. Okay, here we begin with 2N that are replicated. We get N that's replicated, N that's replicated, and then N that's single, N that's single, and single, and single. So we have to have single chromosomes, no matter what in the end. So a similarity would be they both end up with single chromosomes, but these are haploid, these are diploid. These took only one division, this took two divisions. This started diploid, ended diploid, this started diploid, ended haploid. And there's your differences between mitosis and meiosis. And finally, now this is just more differences, okay, in a written form. You can look that over. And finally, how did we get all this variety, okay? So here's talking about that independent assortment. Remember, here's where we see both the daddy ones line up on one side and the mommy ones on the other side. We see sperm that carries nothing but daddy genes, sperm that carry nothing but mommy genes. But here's another possibility. If a daddy thing comes over here and a daddy thing on the other side, then we have daddy and mommy things in these sperm, daddy and mommy things in these sperm. That's independent assortment. That's one way that we can get variety. The other variety, you guessed it, was the crossing over, the little switcheroo. And here it is up closer, okay? There's that word, chiasma, okay? Excuse me. Okay, the chiasma is where the actual crossing over is occurring. And you can actually see that under a microscope. It's pretty cool if you ever find a cell that's like doing that. But you would have to be looking in the ovaries or the testes if it was a human. Okay, but you can see where they did a little exchange. So not only would the independent assortment bring about a lot of diversity, but so would this. Now we see a whole daddy gene here, but a daddy gene that has a little bit of mommy on it, a mommy gene that has a little bit of daddy one, and then a whole mommy gene. So we get more variety. And variety is what continues with the species. But there, we're done. All the videos on this. One more time, let's take a look at the outline, okay, to see what we've accomplished here with all these videos. If you stayed awake through all that, we just finished up talking about sexual reproduction and meiosis. We talked about the divisions and the pairing of the homologous chromosomes, very important. We talked about independent assortment and crossing over, but we didn't mention two other forms of genetic variation and where they come from. Cover up this part really quick. The two other forms are, there are 7.6 billion humans in this world. Don't you ever wonder who you're going to get married to? You know, I remember thinking when I was younger, like, who am I going to get married to? But that one person in 7.6 billion, your child is going to be that 1 in 7.6 billion chance that you two got together. That's just the beginning of the diversity, because there's so many possibility of people that you'll meet out there and have a family with someday, or not if you choose. That's the beauty of freedom. Okay, independent assortment, we said how that diversity can come about. There's actually a mathematical equation that you can do to figure out like how much. You go 2 to the x, where x is the haploid number. So like in humans, it'd be 2 to the 23rd. It shows all the different possibilities of independent assortment. So that's like 2 times 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 2. Okay, so 23 times, you got to multiply by 2. Crossing over, we see how that can come up, but also random fertilization. Guys, you produce a lot of sperm. Okay, there are millions of little sperm in there. The one that gets through is quite the lucky fellow or feller. Okay, and it's the one, and again, that's a mathematical equation. You go 2x, x being n, 2 to the 23rd times 2 to the 23rd, and that's the possibility of which sperm will fertilize which egg. Okay, so just if you kind of multiply the possibility of humans out there, 7.6 billion, times this number that you figure out, times the possibility of crossing overs, and we'd have no mathematical equation for that because it's unlimited. I'm sure it's limited, but I don't know if there's a mathematical equation. I doubt it. They would have to figure out how many genes are out there, but also the probability of crossing over. And then the random fertilization, multiply all these numbers together. So you are one lucky person when you think about it. The chances that you became you, genetically you, and nobody else are just one in like probably more than a Google. Okay, and that makes you very special. Okay, and to talk about an evolution connection, oh well, this diversity just basically leads to more diversity leads to better evolution, being evolved, best fit for our environment. Okay, and that's it for that. Now you're probably wondering, what's all this? This stuff here in the outline, this is all the crosses mainly. There's Mendel and his little laws and stuff. Then these are all the crosses. These are the crosses that we did. Remember, I will definitely be going over the multi-hybrids, dihybrids. I'll also be going over epistasis. Pleiotropy, I'll mention that word. Polygenic, I'll go over that as well. Put a little X next to that. But you should remember how to do complete dominance, incomplete codominance stuff, multiple alleles, like that's like blood type is a good example of that. You should know how to do the sex linked ones. Pedigrees, those are little charts with the circles and the squares. I'll be giving you some of those to see how you are. We talked about disorders already. Chromosome theory, we'll talk about that a little bit, and we'll be done this unit. Okay. We are trying to go through this unit fast, but really at any time